try to start that now. Okay, I think we're recording, so that's great. Thanks everybody. My name is Ted Abel. I'm at the University of Iowa. And what I want to talk about today are some findings that we've had, some really findings in basic neuroscience that have really, we think, connected to studies of neurodegeneration and maybe even potentially the development of some additional and therapeutic approaches. So the work in my lab focuses on gene expression, and the work I'll talk about. And experience induces gene expression in particular circuits of neurons. In classic work shown here from John Guzowski, Paul Worley, and Carol Barnes, they looked at an immediate early gene called ARC, and they found that it was induced in particular sets of neurons here in hippocampal area CA1, where you can see the red induction. We now think of these as quote unquote engram cells, and as a way to both store and retrieve memories. Now, my work has focused on this gene expression in the context of memory storage, but also in the context of sleep, and then in mouse models for both neurodevelopmental and neurodegenerative disorders, which I'll talk about today. The only part of my lab really I won't talk about today is our work on sleep and our work on autism. Happy to take any sorts of questions at any time. Please either jump in by unmuting yourself or put a note in the chat, and I'll try to get to it as soon as possible. Now, historically, the gene that has been the initial focus of gene expression with long-term memory has been the protein CREB, that is cyclic AMP response element binding protein. What was interesting about CREB in the late 80s and 90s was that it was a molecular switch to activate gene expression, that it was present at rest in neurons, and that it was activated by phosphorylation by a variety of kinases, all of which are activated by neuronal activity. Kinases ranging from protein kinase A to calmodulin-dependent kinases to MAP kinases. Now, what became clear in the early 90s is that in the late 90s, rather by a number of groups, Michael Greenberg, Hilmar Batting, Anirvan Ghosh, Dick Chen among them, was that CREB phosphorylation was not sufficient to drive a gene expression. And here's an example of one of these studies in culture that I think is particularly informative. And in this work, neurons were exposed to different levels of glutamate. And in the words of the Sesame Street song, one of these things is not like the other, which is to say that you get CREB phosphorylation here on the left at both concentrations of glutamate, but only CREB-driven gene expression at the higher concentrations of glutamate. So this suggested to many of us in the field that there were other signals that were needed. We now believe that those other signals are co-activators. They are transcriptional co-activators recruited by CREB and recruited by phospho-CREB that then regulate gene expression. So one of these is CREB binding protein, or CBP. That's closely related to a molecule called P300. And another related molecule is CRTC, or TORC, which is CREB-regulated transcriptional co-activator that binds to the B-zip domain of CREB. In general, with transcription, it's regulated by co-activators and co-repressors, and they get recruited. For example, CREB binding protein binds some 250 transcription factors, but in particular, it's responsive to CREB phosphorylation. It then drives target gene expression. Now, what's striking about CREB binding protein is that it is a histone acetyltransferase. It binds all these proteins, but it also has an enzymatic activity, and that enzymatic activity acetylates the N-terminal tails of histones that are present in chromatin. A reverse enzyme called histone deacetylase takes off those acetyl groups and then drives a more repressive state, a process in memory that has been studied by a large number of labs since it was first identified in memory. Actually, the first study goes way back to the 70s, but the more modern studies are in the mid-2000s. Now, in our own work, from Marcelo Wood, who is now a chair of neurobiology at the University of California, Irvine. Marcelo looked at several mutants of CREB binding protein. One particularly informative one was CBP KIX, generated by Paul Brindle when he was at St. Jude Children's Hospital. And here, the kinase-inducible binding domain of CBP is mutated, a triple point mutation. And that gives rise to memory deficits. And so what's striking about these memory deficits, here shown for contextual fear conditioning, is that they only show up after during a long-term memory test. So short-term memory is normal in these animals, but long-term memory is impaired, and selectively long-term memory for context is impaired. Now, reciprocally, we can enhance memory by blocking these HDACs. This is work by Chris Vesey, who is now at Skidmore College in upstate New York as a tenured associate professor, as well as Marcelo. And we used an inhibitor. One of them is trichostatin A. There are several others that block HDACs and therefore drive the acetyl state and lead to enhancements in fear conditioning. So what's particularly striking here is that we can get reciprocal impairments of memory and then enhancements of memory by manipulating acetylation in different directions. Now, my lab sought to identify the target genes that would mediate this enhancement of memory. And that's really how we've gotten to this whole line of work that I'm going to focus on today in our paper that was published in Science Advances last spring. This work here was work using CREB mutant mice carried out by Chris Vesey and Marcelo Wood. And what they found was that trichostatin A, this HDAC inhibitor, would not enhance memory if CREB was mutated. Wild-type animals, you get an enhancement at a low and high dose, but in CREB mutants, you do not. And what they did was to look at what genes would be regulated by histone deacetylases, by histone acetylation, after memory, but not regulated in CREB mutants that do not show memory enhancement. And they identified two genes that are members of a subfamily. Those genes are called NR4A1 and NR4A2. I'm going to be talking about them for quite a lot of my talk. The NR4A here stands for nuclear receptor. So they are a member of a large family of nuclear receptor genes. They are so-called orphan nuclear receptors because unlike estrogen receptors or progesterone receptors, they don't have known ligands. They have a variety of names and have been studied as immediate early genes like ARC or FOS for a long time. NR4A1 is also called NGFIB or NER77. NR4A2 is often better known as NER1 and NR4A3 as NOR1. These are ligand-independent transcriptional activators. They heterodimerize and homodimerize, interestingly, with RXRs. And they've been linked to cognitive deficits and schizophrenia in mice and humans. And I should also say Parkinson's disease. NER1 or NR4A2 is a marker of dopaminergic neurons in the midbrain and is critical for their development and also then their degradation in Parkinson's disease. Now, Josh Hawk, when he was a grad student in the lab, worked with David Mangelsdorf's lab to look at the superfamily of nuclear hormone receptors. So this is the largest family of transcription factors in metazoans, and there are some 40 members or so. And all the ones here shown in red are ones that are upregulated after learning. And you'll see NR4A1, 2, and 3 here. You'll also see some other interesting things like PPAR gamma and RXR alpha and LXR. Interestingly enough, a number of these drugs like pioglitazole and others that are agonists of these compounds serve as cognitive enhancers, and so are very interesting compounds. NR4A1 and 2 are upregulated in a very specific time course after learning, rapidly within an hour, and then come back to baseline. Now, sorry. We have looked in unpublished data at in chromatin changes after learning, here using single nuclear ATAC-seq in work that Jyoti Chatterjee and Jan van Robes in my lab have done. Jyoti earlier this week started his own lab as an assistant professor at the University of Iowa. And I should say, if any folks are looking for postdoctoral positions or research scientist positions or graduate students positions. Both my lab and Jyoti's lab are both looking for people to work on this project I'm describing today. So what Jyoti and Jan did was to carry out single-cell ATAC-seq and then to look at the NR4A1 and NR4A2 promoters. And you can identify that promoter by CBP chip-seq data and see that at the NR4A1 promoter and the NR4A2 promoter, there are more reads in the ATAC-seq sequencing after conditioning than in the baseline. And that's found particularly in the subset of nuclei that are CA1 nuclei. This is actually a combination multi-omic experiment where we look at both gene expression and ATAC-seq. Sorry, excuse me. Now, how can we study the function of this family? Now, one of the challenges here is you have a family here with these 3 members, and so that all which bind similar sites. So what we did was to take an approach of making a dominant negative. Josh Hawk did when he was in the lab, and he made a dominant negative actually that came from work in T cells, actually, where NR4A factors are also expressed and drive immunity and immune responses. And he substituted the YFP for the activation domain, and that generates a protein that binds DNA but then doesn't activate transcription. These NR4A dominant negative animals have memory deficits. Here in work from Jyoti Chatterjee, this is looking at object tasks where we do spatial object location memory, where we train animals with 3 objects in certain places in space and then move one, here the circular object, and animals will explore this moved object more than the other objects if they remember that it was moved. If they know that it was moved, they hence remember its previous location. And see, wild type animals exhibit that selection for this moved object, but the NR4A dominant negative animals do not. Marcelo Wood has done a number of studies of this family, and here's some of his data using siRNAs to NR4A1 or 2, showing that they also see deficits. Now, what's interesting is that we can overexpress or activate these transcription factors in aged animals. And this is work from Janine Quappas and Marcelo Wood on the left here, where they virally overexpressed NR4A1 or 2, and were able to enhance memory in this object location memory paradigm. In our own work from Jyoti's work in 2020, using an agonist developed by Stephen Safe at Texas A&M called CDIM12, an agonist of the NR4A family, Jyoti found that he could enhance memory here shown on the right in aged animals. These are 1-year-old mice, I believe, in both of these studies. Now, what about links to disease? And so this is where we get to what's of interest here and to what's in our recent paper. So what Jyoti did with Ethan Ball and Jake Michelson here is look at the data in the Allen Brain Institute's database of gene expression in human ADRD patients, and found that levels of NR4A 1, 2, and 3 were negatively correlated with SERAD scores. So as the severity of disease increases, levels of these transcription factors decrease. Similarly, in a tau model of Alzheimer's disease and related dementias, this P301L model, RTG4510, Jyoti found that those animals had deficits in spatial memory that also had reductions in NR4A 1 and 2 here in the upper right-hand corner. One thing I want to note, many people criticize this tau model because it's a gene insertion model, and there are several genes that are mutated by the insertion. So we included a control here where we gave animals doxycycline. Doxycycline blocks the expression of the tau transgene, does not change the genomic insertion of the transgene. And these animals do not have memory deficits. So people criticize this animal and they say it's confounded by the mutation of genes near the transgenic insertion. However, we think that it is an appropriate model because when we block transgene expression, we bring back memory. Okay, so I think this is a very important study that's kind of buried in our paper. that I think people get all hot and bothered about these mice. Every mouse model is going to be complicated. The nice thing about this model is you have a conditional regulation with doxycycline, in my mind. Now, importantly, what Jyoti found was that he could take a viral expression approach like Janine Quapis and Marcelo Wood did and overexpress NR4A1 and rescue memory in these tau models here in the right-hand panel shown in purple. So what I've told you about so far is sort of a very long story that we've been studying since the early 2000s that goes from Krebs phosphorylation to histone acetylation to gene targets of these nuclear receptors, NR4A1 and 2, to cognitive enhancement in aged animals to a cognitive enhancement in tau models. Now, everything I've told you about is in the nucleus, and it has to do with transcription factors. How do we get to the synapse, and how do we begin to study what's going on at all of these synapses that are formed between neurons in the brain? Now, to do this, Jyoti did an experiment where he did RNA-seq, but there's a twist that he did. He waited 2 hours after training to do this sequencing. Now, the interesting thing about that is that allows all the immediate early genes that we sort of know about to be rapidly expressed. And then we're looking at these somewhat later genes. And he did this by looking at NR4A dominant negative mice, which have memory deficits, and compared them to control mice. Excuse me. And what he found was that there were a set of genes that were down-regulated in these NR4A dominant negative mice relative to control mice. And when he looked at them, he found that they all fell in related pathways. So here's a cluster of these 40 genes. And you'll notice here that all of these genes are related to disulfide folding and chaperone binding and protein processing in the endoplasmic reticulum, as well as this unfolded protein response. And so these all cluster within the endoplasmic reticulum. Let me take a break. I just saw there was a question. Thank you for that question. And that had to do with the doxycycline treatment. And it's a good question. And Tina asked it about doxycycline like minocycline have neuroprotective and anti-inflammatory impacts. Did you have doxycycline controls apart from gene modifications? We didn't do that in this study, but we've done it in aging, and we haven't seen an effect of doxycycline. We only gave doxycycline for a couple of weeks here to suppress expression. So it's a good point, Tina, and we have actually not done the direct control in these mice. Thank you for that question. It's a good question. Thanks very much. Okay, so the important thing here in terms of what Jyoti has found is that there's a cluster of genes that are regulated by NR4A, we think, although we have to show that directly, and that regulate protein processing and in the endoplasmic reticulum. Now, what's striking about this is that we talk a lot about gene induction after training and the induction of protein synthesis, but very few people think about the fact that you have to process all those proteins that are synthesized during memory consolidation. And so we think that this is naturally doing that. A lot of people think about this as the unfolded protein response, the misfolded protein response. But this is actually also the protein folding response. This is the way in which membrane proteins are processed to be presented on the surface of the cell. Here's another way to look at this with another string package. You can see that these regulated genes, a lot of them all fall together and cluster together. And NR4A and the NR4A chip experiment binds to promoter regions of HSPA5 and PDIA6, two of the genes we're interested in. And in NR4A dominant negative mice, this is a separate group of animals as a control, we see reductions in both the mRNA and the protein. I should note that this protein is carried out in synaptosomes, so it shows that this HSPA5 protein is present in ER that are in synaptosomes. Now, in work that is submitted but up on bioRxiv, we have seen that we see the signature of these endoplasmic reticulum folding genes in spatial transcriptomic data. This is spatial transcriptomic data using Visium, a 10x genomics platform, where we see gene activation in the cortex as well as CA1 after SOR. And you'll see the genes that I've been mentioning, these HSPA5s, the DNAJs, you'll see them here in this kind of greenish sort of turquoise color, as well as what people think of as more conventional transcription factors like FOS and NR4A2 that are also induced. So what about the NR4A dominant negative mice? Can we come in and rescue them by changing levels of HSPA5, one of these chaperone target genes? So what Jyoti did was to create a virus that expresses HSPA5. He just expressed this for a couple of weeks to try to get endogenous levels of HSPA5 instead of overexpressing it. And what he found here in the bottom left here is that there's an increase in memory in NR4A dominant negative mice that overexpress HSPA5. We can chemically induce chaperone activity with enzyme PB with a chemical PBA, a related molecule, a sodium butyrate does not act in that fashion. And this chemical chaperone also leads to enhancements in long-term memory in the NR4A dominant negative mice. Now, what about the downstream targets of these folded? What proteins are folded by this complex of chaperones? That's very much where we are focused now in my lab. And we looked at one candidate molecule that's been identified in the literature, and that's GluN2A. GluN2A is NMDA receptor subunit 2A, and its surface presentation is dependent on this endoplasmic reticulum processing. And so what we did, what Utsav Mukherjee did in cell culture, is to look at surface GluN2A in hippocampal neurons and cultures that have been depolarized using KCL. And in stimulated neurons, he sees an increase in the surface levels of GluN2A. Those are not seen in NR4A dominant negative neurons that express a dominant negative for NR4A2. These co-localize with PSD95 here in the middle panel. And in the right-hand panel is a panel where Utsav overexpressed HSPA5 in the presence of NR4A dominant negative and found that he could increase the surface levels of GluN2A at the synapse by giving HSPA5 to these mutant, to the cells that have NR4A2 blocked. So what this argues is that NR4A2 and NR4A transcription factors act to regulate the protein chaperone machinery that includes HSPA5. And by providing that chaperone machinery, we can restore the surface presentation of this NMDA receptor. And so what I've told you today is, and hopefully we have some time for some discussion, I tried to truncate this a little bit, is a process that really starts at the synapse with calcium influx through NMDA receptors, leads to gene expression through CREB and CREB binding protein. I didn't talk about this source of acetyl-CoA, ACSS2, from our work with Shelley Berger, but I focused on this targets of NR4A 1 and 2, and then their regulation of endoplasmic reticulum genes, and then the surface folding and presentation of clientele like the NMDA receptor GluN2A. Obviously, there are a lot of questions of how this might apply to additional models of neurodegenerative diseases. We've just looked at some experiments in tau models, but Jyoti in his lab is going to be looking at APP and PS1 models, and then trying to look at the role of both the NR4A1 transcription factors as well as some of these chaperones in those cognitive deficits that are seen in those animals. So let me really first thank Jyoti Chatterjee here, who as a postdoctoral fellow and research scientist drove this work, Jan van Robes, who's our bioinformatician, who's helped with all the data analysis, and Utsav Mukherjee, who carried out the cell culture work that I've told you about, and a variety of funding sources, particularly here, funding from the National Institute on Aging and the National Institute of Mental Health for their funding of our work on epigenetic mechanisms of memory storage. And we'd love to have you, if anyone's interested in coming to join us at Iowa or following me on Twitter. I'm still on Twitter. We talked about that beforehand of whether we should still be on Twitter or not, but that's at Ted Abel Neuro, also LinkedIn and Facebook as well. So let me stop sharing, and I don't know if you want to. Do we want to stop the recording?
You can if you want. That makes it easier to upload. So
okay, I'll stop the